Thank you all for joining, and I will now introduce our presenter for today. Dr. Jonathan Gutman received his bachelor's in history from Princeton University. He completed medical school at New York University, residency at the University of Colorado, and fellowship at the Fred Hutchinson Cancer Research Center. He returned to the University of Colorado in 2009 and currently serves as a medical director of cellular therapy, is the clinical medical director of the University of Colorado Stem Cell Lab, and is the medical director of the Gates Biomanufacturing Facility. His research focuses on optimization of allogeneic transplantation and immunotherapy, with a particular emphasis on cord blood transplantation. Thank you, Dr. Gutman, for being here with us today. I will stop sharing and turn the floor over to you.
All right, thanks very much, Victoria. I'm going to go ahead and try and share my screen. Let's see if we can find the deal. I'm going to blow this up right now. All right, are we looking good on the screen?
Looks great.
Okay, great. Well, welcome everybody, and thanks for being here. Kind of wish we could be in person. I rather kind of work the room, but we'll deal with the Zoom if we have to. Just as a quick note, I gave Department of Medicine grand rounds on this topic about a month ago, and what I'm going to talk about today is largely similar. So if you happened to be there, you might just want to tune out and collect your CME here, because the talk is fairly similar. For those of you who I don't know, first of all, I just would like to say thank you to all that you do for us and for our teams and for advancing cellular therapy along with everything else. But I direct the bone marrow stem cell transplant program and have for a long time here. And this whole world of cellular therapy has emerged over the last decade or so, and it's sort of a natural fit for folks who did transplants previously. And so I want to talk today about sort of what it is and where we are and where we're going with it. So why don't we just go ahead and dive in? I think hopefully an interesting and exciting topic to folks, as it's pretty cool, cutting edge stuff that's really starting to have an impact on patients. So I like to start the discussion by just taking a minute to talk about the case of Emily Whitehead. Don't know if any of you guys are familiar with her, but she, in April of 2012, I think was 6 years old, was the first pediatric patient to get these new therapies, this new therapy that we called CAR-T therapy at CHOP, the Children's Hospital at the University of Pennsylvania. She, I think the sort of the story has got a couple layers to it, and it's so good that I really like to tell. Is such a good starting point. So she had refractory acute lymphoblastic leukemia, ALL, which is a disease that we've made great strides in treating, and the vast majority of pediatric patients who get this disease can now be cured with conventional chemotherapy. But with those small portion of patients who do relapse or are refractory to initial therapy, disease is a horrible disease, and options have been very limited. And so Emily, back in 2012, was, teeming with leukemia, really had no options, probably within days of dying of the disease, and she was the first kid to get infused these things that we call the CAR-T cells. And so they infused these cells into her, and that night she just got unbelievably sick. Her temperature went well above 40 degrees. Her lungs were filling with fluid, intensive care unit pressors. And the added layer of the story that I really like, particularly when we start talking about our rheumatology colleagues who are starting to enter into this space, is that she was unbelievably sick. Nobody quite knew what to do. And as the story goes, I think they may have run a panel of cytokines on her and saw that one of these inflammatory markers, IL-6, was incredibly elevated in her system. And it turns out that one of the fathers of CAR-T, who was who had developed the product that Emily received, had a kid who had juvenile rheumatoid arthritis. Carl June's his name. And his kid was at the time on this very new drug called tocilizumab, which is an IL-6 inhibitor, which I think we're all familiar with now. Was used in COVID and whatever, and sort of with nothing better to do, they decided, let's try and give this drug to Emily and see what happens. And that night, she calmed down after they gave the drug. Everything sort of came back under control, and that's sort of where the story begins. And you know, she's now actually 12 years cancer-free, and a poster child for this new therapy, but really just a remarkable story, and one of these moments in medicine where, again, you don't need statistics to tell you that you've had an important impact. And that's really where the story begins on these cellular therapies in the modern era. And so I just think it's just such a great story and a good place to start. So what I'd like to do today is talk a bit about the current state of cellular therapy. What is it? What are the modalities that we talk about? And then I want to talk a little bit about our institutional contributions to the field, which many of you, I think, helpfully were involved in, and think a little bit about what the future might look like as we consider operational issues around how we deal with these types of therapies, which are coming fast and furious and are quite complicated. And the modality I think that people are most familiar with is what we would call CAR T cells, but there's a variety of others, and we'll talk about them. And they're being used in an increasingly broad set of disease states. Much of this work has started in blood cancer and is most developed in blood cancer, but making great progress in solid organ malignancies and other autoimmune diseases. So we'll walk through some of those things today. I think a couple of important things to bear in mind as we think about what the future looks like in this space and how we think about organizing it. And you know, I think some of these organizational questions are relevant to you guys working in the research fields as we think about how to organize our infrastructure to support these studies. But a couple of key points that I think are worth bearing in mind is that this is a field that's very, very rapidly evolving. I just add to I had on the last slide, didn't dig into it, but generally speaking, I think when we think of cell therapies, we're talking about living cells that we're going to use for some sort of therapeutic benefit, perhaps after some degree of manipulation. A little bit different from what we would call gene therapy, which is sort of a sister thing that's also coming down the pike. Gene therapy, I think of as maybe being more about inserting a gene into somebody's cells to have it constitutively produce some sort of thing that's missing or needed. There are similarities between the fields, and we're starting to get organized around gene therapy. That will be coming down the pike too, but really focused today more on cell therapies. But for both of those fields, it's true, they're very rapidly evolving and things are changing very quickly. And what we're doing today is probably not what we're going to be doing 5 years from now. And so I think we always need to be bearing that in mind in the back of our heads as we're thinking about what's going on, because it's easy to kind of get trapped into thinking, well, now, you know, we know how to do this and this is what it looks like. It's all changing fast. And the different therapies we're using and the different diseases involved and the different stakeholders are all expanding. There are competing therapies. What I call bispecific and trispecific T cell engagers would be a good example of something competing with these cell therapy, with CAR T cells, where we just use basically like a small molecule to try and have the same impact. And so all this stuff is in rapid development. And so any operational system that we create needs to be adaptable, it needs to be flexible. And people always need to bear in mind that what they know today about something may not be applicable to what the next therapy looks like. And it's just, so it makes it very exciting, but it's also got a lot of unknown associated with it. I would say too, when we think about operational questions around these therapies, important to understand that some of them are now FDA approved. And when something becomes FDA approved, the operational issues associated with it become quite different than the issues in clinical trials. And I know you guys are kind of largely the clinical research folks, so maybe less interested in some of those details, but happy to flesh them out as needed. But important to bear in mind that once it becomes approved, it's sort of a different deal in terms of a lot of regulatory and other issues we have to think about. All right, so we can start out with what I am not like some sort of huge nucleids on the block fan, but I think of CAR T these days as being appropriate to sort of think of as the old new kids on the block in this space. Pretty, cutting edge, but at this point, a little bit old news to those who've been doing this for a while. So let's walk through what is a CAR T cell and how does this work? So CAR stands for chimeric antigen receptor. And what we do with a CAR T cell is we take a patient's own T cells generally, although we're also working on using shelf products for this, but these days, all the approved products involve collecting a patient's own cells, and we take their T cells and we use a viral vector to insert into those T cells the genes to code for a receptor that the T cell will express on its surface that we can then target to whatever we build this target against. And then there's a couple of additional components to the CAR T molecule. Most importantly, there's what's called a co-stimulatory molecule. There need to be some other pieces attached that help to stimulate that T cell. And different molecules that you use as a co-stimulatory molecule, we've come to understand will give CAR T cells different characteristics. But the general idea is that we use a virus to insert the coding into the T cells for them to produce a receptor that we will then use to attack whatever it is that receptor targets. So we'll flesh this out a little bit more, but that's the general notion of how a CAR T works. Now, CAR T therapy, importantly, is complicated stuff. The process of doing it, at least as we do it now for the vast majority of the CAR T cells, involves having to meet the patient, make sure that they have the resources necessary to be able to get through this whole process, which includes evaluations by variety of team members, including the medical evaluation. We will then collect the patient's cells, process we call apheresis or leukapheresis. And then we will take those cells and we will ship them to a manufacturer, whether it be on our campus or an industry sponsor who's doing this. The manufacturer will then produce the CAR T cells, which generally is about a 2 to 3.5 week process. Then we will bring the patient in. We will administer lymphodepleting chemotherapy. So chemotherapy designed to sort of knock down their lymphocytes, usually fludarabine and cyclophosphamide. And the main reason to do that is that it then creates a cytokine milieu where the body is producing all these cytokines to cause lymphocytes to start expanding and growing. And when we put the CAR Ts in, we want them in that environment so that they'll get growing and do their thing. And then we have to monitor the patients. Typically for a period of at least 28 days, they have to be local to be monitored for adverse events and issues. Increasingly, we're able to do much of this outpatient, but sometimes also needs to be done inpatient. And these are often very sick patients for leukemias, for lymphomas that are getting to CAR T cells. They're often super sick and they don't have a lot of time, and so we have to do this quickly. They may need chemotherapy between leukapheresis and the time of the CAR T to hold the disease at bay until we can get the CAR Ts into them. But that's the general process, and it's a fair ado. Again, these are a living drug. We put these cells into the patient and they effectively act like normal immune cells, and you have an immune response similar to what you would see if you had a virus or some other thing. And so what we want to see is we put the cells in after the lymphodepletion, and over that first week and a half or so, they expand up into considerably larger numbers as they're growing and responding against their target. And then they tend to back down, just as happens in other physiologic immune responses. And then, generally speaking, hopefully we see them persist at a low level in the blood system, but we'll come back to that a bit. But that's sort of what happens with these CAR Ts when they go in. So as of today, the vast majority of work that's led to FDA approval has again been in the space of blood cancers. And I just highlight here some of the registration studies, the studies that led to the approval of these agents in the various diseases in which they're approved. And the main diseases for which we use them now are acute lymphoblastic leukemia. Actually got started first in the pediatric population, one of the only places where peds came before adults, I think, in terms of a drug getting approved. And then we've extended their use to non-Hodgkin's lymphoma, particularly diffuse large B-cell lymphoma. And most recently, we've been using them also and have 2 FDA-approved agents for multiple myeloma. And the list of these registration studies is sort of it's just a laundry list of New England Journal of Medicine papers or Nature Medicine, Lancet, the biggest journals we have in the field. And so, if you follow this, almost every week there's a new CAR T paper in these great or new cell therapy paper in these great journals, just reflective of this growing exciting field. And I think if you look at the data here, what you'll see is that the response rates in these patients are fairly extraordinary, 80-90% response rates. And in these registration trials, these are very heavily pretreated patients who who had no major alternatives. And I think it's fair to say, though there's details around all of this, that probably 30 to 40% of patients who otherwise were guaranteed to die are being cured by these CAR T therapies across the board. So that's a pretty big step up when you think about things in the world of medicine. And if we think about the agents that are FDA approved, this is actually current as of October 2024, not September 2024. There are now 6 of them, 4 of them for lymphoma and ALL, and 2 of them for multiple myeloma. And there's some details that are different about them, but they're generally relatively comparable. But what I would say is perhaps sort of most important as we think about the immediate future around these issues. I've highlighted in red recent approvals, which are, generally speaking, involve moving these therapies earlier into lines of therapy. So, as you well know, I think the paradigm for drug development is generally that we'll start with giving the drugs to patients who are super sick and have failed everything, and we test their safety first. But then if they look good, we start moving them earlier and earlier into therapy. And that's exactly what's happening with CAR Ts right now. They, for diffuse large B-cell lymphoma, we now can administer the CAR Ts after the failure of 2 lines of therapy. In multiple myeloma, one of the agents, Carvykti, is now approved after just 1 line of therapy. And that has a lot of implications for volumes and for some financial issues that we'll talk about later too. But these things are proceeding as one might expect, earlier into treatment, and hopefully earlier in treatment will be better tolerated and also more effective. So when we think about why the CAR Ts don't work in those patients where they don't work, there's generally felt to be 2 kind of major reasons why a CAR T might fail a patient. And if you look at one of the early 2018 papers, this was on BALL in children, at the pattern of how patients relapse, you can kind of see there's sort of 2 steps here, one sort of in this 2 to 4 month period and one more in the 8 to 10 month period, where it looks like patients are relapsing who relapse. And it's generally felt that there's sort of 2 major issues going on here. The early relapses may occur because for whatever reason we put the CAR Ts in and they just don't expand well. They kind of just fizzle out and go away. And when we see that happening in patients, generally speaking, it's a harbinger for likelihood of relapse. And so that's one area we're working on. But then the later relapses, we think, generally occur because the diseases develop resistance against the target for which the CAR Ts are designed to attack. And so there are strategies which we'll talk about to try and get around that issue. Should just mention on the targets, The primary target that most everybody has talked about historically is something called CD19. CD19 is expressed on B cells, and so the CAR Ts will take out all the B cells along with bad cells. But CD19 is expressed on B cells, so it's expressed on B cell ALL and B cell non-Hodgkin's lymphomas, and is a good target for those. For multiple myeloma, the target that we've targeted is something called BCMA, which is expressed on plasma cells, which are the cells that are the cells of origin for multiple myeloma. Picking an appropriate target is one of the great challenges in CAR T therapy, because you have to pick something that hopefully you can just hit that and not have collateral damage and be doing damage to all kinds of other targets, but it also has to be something that's on the bad cells. And so picking those correct targets is a challenge. As we'll talk about in the context of solid tumor oncology, there's a bunch going on in that space. So when we think about the toxicities associated with these therapies, so classic CAR Ts, really the CD19 CAR T, has two major toxicities associated with it. But these are toxicities that I think certainly the first would be reasonably extrapolated to likely occur in other settings also. The first issue we see is this thing called CRS, cytokine release syndrome. And the second that we see is neurotoxicity. And I'll flesh those out a little bit further. But there are some other things that are starting to emerge as issues and potential concerns among CAR Ts. They are going to have expected toxicities. The CD19 CARs will cause B cell aplasia. We will destroy a person's B cells. So we need to give them immunoglobulins to make up for those lost B cells. They may cause a tumor lysis syndrome if you put them into a patient who has a lot of disease and then start killing the disease. So that needs to be monitored. With the BCMA CAR, that's the CAR for multiple myeloma, there seems to be a hint emerging that later down the line, more like months after the infusion of cells, that some patients may start to develop Parkinsonian type symptoms. So we need to watch that very closely, because we've got to be careful with what we're doing here. It's also true that in some of the CAR Ts, and particularly in CAR Ts targeting a molecule called CD22, which is another B cell molecule, this what we call HLH or macrophage activation syndrome, which is generally sort of a just profound inflammatory response in the system, is emerging as a potential toxicity. We actually had a patient on one of our trials die from this. This generally occurs about 3 weeks to a month after the transplant. So that's something that we need to be on the lookout for. These patients are vulnerable to infections, need supportive care there. But a final major consideration, which I'll comment on a little further because it's received some attention recently, is the possibility, anytime you do any genetic manipulation of cells, of what we would call insertional mutagenesis, the possibility of basically causing a cancer by inserting genes into normal healthy cells. And that's a major issue in gene therapy, but we'll talk about it in the context of CAR T also. But the two big sort of classics are cytokine release syndrome and neurotoxicity. And generally speaking, cytokine release syndrome is just the acute inflammatory response that occurs as the cytokines are being released from these cells that are going in and doing their thing. And we generally see it as an issue roughly 3 days to the first week or so after the infusion of cells. The neurotoxicity issue, it may be a little bit more of a CD19 specific issue, but it tends to occur a little later, 1.5 to 2 weeks after the infusion of the cells. And we'll talk now about the two different syndromes. So cytokine release syndrome, again, is sort of this just immune flare. Patients will get with it high fevers, hypotension, hypoxia, and we can grade it based on the severity. But grade 3-4 patients may end up in the ICU on pressors, super high fevers, conceivably needing to be intubated. The incidence of it occurring varies a bit with the different CAR T you're using, the burden of the disease. We're getting a better understanding of it and getting better at dealing with it, but it's fairly variable. But these days, I think the incidence of our seeing severe grade 3-4 toxicities is quite low, in the single-digit percentage point range. But it absolutely happens, and lower grade 2 is very, very common. General management is tocilizumab and steroids. there are a number of agents that we're investigating also as potential therapeutics, all sort of anti-inflammatory in nature. The other big side effect issue that we get concerned about is neurotoxicity. And here, this is sort of a strange thing. It can sort of manifest in almost any way. People, you can see seizures, you can see word finding deficits, you can see aphasia, you can see encephalopathy, you can see motor symptoms, you can see elevated intracranial pressure. And similar to cytokine release syndrome, we have a grading system based on the severity of symptoms. And the incidence is relatively similar to that of cytokine release, maybe a little bit lower, but influenced a bit by costimulatory prior treatment, whatever. But definitely an area that we continue to actively investigate. May have something to do with CD9 receptors in the CNS that are being attacked, but I don't think anyone really understands it well. And its management tends to be more supportive care than anything else. We can give steroids, are looking at some investigational anti-inflammatory agents. Tends to get better, but it can be a scary thing, and it can be quite profound. So that's the neurotoxicity issue. Now, I mentioned also the potential concern about triggering a malignancy by inserting these genes into these T cells. And this, again, in gene therapy, this has been a significant issue. Patients getting gene therapy for sickle cell, for hemoglobinopathies, for hemophilia, other inborn errors of metabolism. There have been absolutely case reports of leukemias developing in those patients secondary to the gene therapy effort. So it's a serious issue with potential concern, and it kind of got a lot of press earlier or late last year when really out of the blue on November 28th of last year, the FDA announced that it was investigating the serious risk of T-cell malignancy following CAR T therapy. And that really was a surprise to people in the field. There really wasn't a sense of an issue. And so it turns out that basically among 8,000 patients who were in the FDA follow-up system and presumed 34,000 patients who'd been treated with CAR T, there were 20 reports of possible T-cell related therapies, malignancies associated with the therapy. Now, the details of all of those are not totally known, and there's a lot of complexity in this patient population, but there have been a number of additional follow-up reports around this. The bottom line about it, I would say, is that there's not really clear evidence of a significant concern around the development of T-cell disorders in the aftermath of CAR T-cells at this point. And the incidence of potential reported things is very, very low. One must bear in mind these patients who have these disorders of the lymphatic system already have probably predisposition issues that perhaps could be exacerbated related to the CAR T therapy, but they're not necessarily normal, healthy folks who don't have any abnormalities in their blood forming cells. I will say this thing on the bottom right here is a report using a different mechanism to try to develop the CAR Ts, this piggyback modified strategy. There were actually a couple of reports of what seemed to be clear T-cell lymphomas developing. So the details of how you build the CAR T is important, but the current methodology, I think, generally is felt to be safe and risk-benefit favoring the use of the CAR Ts, but this is something we'll continue to watch closely. All of these patients are expected to be watched for 15 years after the infusion of cells, which some of you in the research world may be stuck having to sort of deal with the monitoring around, which is a little annoying. I will say, too, that I think it's important to bear in mind as we think about risk benefit. If we think about lymphoma, the historic strategy for diffuse large B-cell lymphoma that relapsed would be to try and give the patient high-intensity chemotherapy, do what we call an autologous stem cell transplant, and we would cure a portion of patients with it. But it was known that 5 to 10% of those patients, as a consequence of their stem cell transplant, would end up developing secondary myelodysplastic syndrome, or AML, which is a very bad disease and what you'd have to come to me for an allogeneic transplant. And so, all these therapies carry significant risks, and we're nowhere near an incidence of anything like that in our understanding of CAR T at the moment. Just important, though, I think, to bear in mind. As I mentioned earlier, what's going on a lot in CAR T therapy right now is that the available therapies are moving earlier and earlier into treatment. And on the top here, we have two studies that were instrumental in advancing CAR T therapy in diffuse large B-cell lymphoma, such that it could be used in relapsed refractory patients. These are comparisons of CAR T to the standard historical approach, which would be autologous stem cell transplant. And in both cases, CAR T looks strikingly better. And this transform trial on the left of Breyanzi, some of you may be familiar with, because we as an institution were the lead accruers to the study. Manali Kamdar is the lead author on it, our lymphoma lead. And our HCTU team did a wonderful job on this study. The FDA audited us, of course, and there were no findings. And so that was great and exciting. And so again, opportunity for you guys to pat yourselves on the back about work we're doing here. There's now studies looking at diffuse large B-cell lymphoma in the first line setting in very high risk patients. And then this study here on the right, Carvykti, this advanced Carvykti to second line therapy as it showed clear benefit over alternative standards. So that's a lot of what's going on in the blood cancer space. Now, as we think about the future of CAR T cell therapy, and these principles can generally be applied to any sort of cellular therapy, there are definitely opportunities for improvement. And this wheel circle, if you study it long enough, can help to flesh those things out. But key points I put down here on the right that people are working on include overcoming the resistance potential for these bad cells. Ways to do that might include putting two different CAR receptors on the CAR T, so you're hitting two different places, a variety of strategies in that space. Strategies to increase the potency of the CARs, to have them continue to be highly immunologically active, potentially by inserting genes into them that will help them persist and help them to stay active. Also, particularly in the world of solid organ transplant, the microenvironment around cancers, the local environment can be very immunosuppressive and make it difficult for these CAR Ts to work well. And so strategies to help overcome that are important. Some people are putting suicide switches into the CAR Ts so that you could basically turn them off if they start running amok. So that's something. Different strategies to get them to go to the target that we want more effectively, also being investigated. And then very importantly too, I think the way we do these today, this bespoke process for each individual collecting their cells, producing the CARs, quite complicated, quite expensive. We would love to figure out ways to make that easier and to perhaps develop shelf products that don't have to be generated from the individuals themselves in their investigations in all those spaces, but each of them carry a bunch of challenges also for sure. When we think about the pipeline here, this slide shows number of studies that are open in this space. And you can see the top here, CAR T cells. The majority are in the blood cancer space, but many in solid organ tumors also, and then other areas of disease. And there are hundreds of these studies opened, and they're targeting a whole bunch of different things on the right side. And then there's a variety of different types. There's CAR T, but there's a lot of other types of cell therapies also. And in fact, in the last several months, in the space of solid tumor oncology, and again, we were participants in both of these studies, so thanks to everybody. We have had 2 FDA approvals. So I think of these guys as being the new kids on the block, the young original version of them. But we have a tumor-infiltrating lymphocyte therapy for melanoma and a T-cell receptor therapy in sarcoma that have been approved. So the melanoma therapy, lifileucel, which we are just about to be opening as a standard of care product now, but we were lead accruers on in the original trials. This is a slightly different version of cell therapy, but cell therapy nonetheless. So basically, what happens is a patient goes into the OR with melanoma and has some of their tumor harvested, gets sent to Iovance, the manufacturer, who isolate the T cells that are in the cancer and have the ability to potentially attack it. And the patients are administered lymphodepleting chemotherapy. The lymphocytes are reinfused. They're given high dose IL-2 to stimulate the immune system, rev it up, get them real sick. But these TILs appear to be quite effective in highly refractory populations, not perfect by any stretch, but potentially offering long-term remission and potential cure to a portion of patients. And so they're getting started. And then over on the sarcoma side, we participated in a trial for a rare form of sarcoma, synovial sarcoma, where we use what's called a T cell receptor therapy. So here, what actually is done is a native T cell receptor, the actual T cell receptor that would exist in a patient, none of this engineering stuff. It's slightly manipulated, but a T cell receptor that would target naturally antigens that could be expressed on the sarcoma is basically grown up and inserted into the patient after lymphodepleting chemotherapy. And again, this strategy has had helped outcomes in patients with a very challenging tumor without great options. However, when you use these T cell receptors, you have to rely on the immune system actually functioning as it's designed. So there has to be HLA matching between the T cell receptor that you're targeting and the patient's own HLA. And so HLA, these are the molecules that we look at to match when we do things like stem cell transplant for functional immune interactions. And it turns out that about I think roughly 50% to 50% of people, Caucasian particularly, will have one form of this HLA-A molecule A2. So you need to be HLA-A2 to have this therapy be one for which you're eligible. And then your tumor has to express the antigen MAGE-A4 that it's trying to target. So all comers, that's about 30% of patients with this rare sarcoma. So it's by no means a perfect solution. So there's lots of other stuff going on in the solid organ world, and we have a few trials open here. But the other big thing going on here is that this stuff is spreading into other disease areas. And the place where it's most exciting right now, I would say, is in the autoimmune world. So the idea of using these CAR T therapies to potentially try and knock out autoimmune diseases, particularly diseases that are antibody-based, has been exciting to many. And there have been a number of papers now published around it. And just earlier this year in the New England Journal was reported a case series of 15 patients from the German group that started this with a sort of a potpourri of autoimmune disorders, but very severe, very refractory disorders, and basically all of them getting complete remissions with this CAR T-based therapy. And I think lupus and lupus nephritis are the diseases that have been treated most right now, but all this list of diseases are places that we are interested in exploring. Now, those of you who know me know that I spend most of my energy these days trying to learn about climate change mitigation and the energy transition. And I think that in the autoimmune space right now, there is a bit of a dot-com bubble going on where lots and lots of companies are very interested in entering this space because of this very exciting data that's been generated. But I think there probably are not the number of patients with severe enough disease that need these kinds of therapies, that there are companies going into the space to try and succeed. And so the other thing about me that I waste a lot of time doing is trying to become rich trading stocks in the renewable energy space. And so alongside this sort of picture of all the companies that are trying to do things in the autoimmune space, I've got, 7 different EV manufacturers. And so bonus for anyone who wants to tell me which of these companies each of these EVs represent and which of them have gone bankrupt. And so we shall see about all of that. But I will tell you, it's hard to just get rich trading in that space. So as we think about autoimmune disease as compared to cancers, just some of the issues that I think need to be borne in mind. The risk profile, benefit profile will be different in someone with a bad autoimmune disease than it will be in somebody with acute leukemia. As horrible as the bad autoimmune diseases are, they aren't generally going to be rapidly lethal, horrible in the way that blood cancers are. And so we have to be careful about the potential to cause problems and finding the right patients. Some of the things that seem to be true in blood cancers, like the need for CAR Ts to persist, some of the features for the way that the different co-stimulatory molecules work. I think there are questions about whether those need to be true in autoimmune diseases to get good results. And people ask, well, why do these CAR Ts work better than something like rituximab, which targets CD20 and depletes B cells? And I think there's the feeling that because these are living cells and they are really like harnessing the immune system, the way that the immune system works, I think there is a feeling that that they can get into the tissues and they can deplete B cells in the tissues in ways that these other drugs can't. And this deep tissue B cell depletion may be very important for the effectiveness of the therapies in this space. So the other point that I want to raise about these things and think about just to be aware of is their cost. These are expensive therapies. Those of you who know me also may know that for reasons that I can't entirely explain, I'm actually just about finishing up getting an MBA in health administration to the university. And so these are areas that are sort of interest to me to be thinking about also. But the cost, and this reflects the price that actually is paid, not like we charge $1 million and we get paid $20,000. This is the actual cost of these drugs that the pharmaceutical companies charge. Is, you know, it's $400,000 or $500,000 for the dose. And this list on the left kind of goes in chronological order so that the bottom is the more recent therapies, and you can see the prices going up and up with the more recent therapies. The company manufacturing Yescarta has recently jacked up the price of it. Interestingly, just to give you a sense of how the world works on these things, Kymriah can be used for pediatric acute lymphoblastic leukemia or for adult diffuse large B-cell lymphoma, and it is exactly the same product, whether you use it for one or the other. But because of the potential for greater life years gained in a pediatric patient, they're able to charge $125,000 more for the product. So that's sort of just, you know, an interesting factoid. At scale, when these companies are manufacturing these things, it's costing them roughly $18,000 to $20,000 to make one. So there's a fairly significant markup here. However, you know, pricing of pharmaceuticals is a very complicated thing, and the way our system works is a complicated thing. And so I think interpreting all this is a bit tricky to do, but generally speaking, when we think about whether drugs are reasonable or not, one tool in which you can use is to think about sort of their cost effectiveness, which is generally trying to assess what are you getting for the price that you're paying. The problem with cost effectiveness analyses is that they're highly dependent on the model and the considerations that you put into your model. And factors that can be included there will include how much you expect to cure patients, how long a remission will go, what the disease courses look like, what the alternative therapies and costs look like, what patients' quality of life looks like. And so you can really kind of get your data to look however you want it to look. But there's a list of studies here, if anyone's interested, that sort of try to address cost effectiveness in CAR T cell therapy. And the results really are all over the map. Generally speaking, people think that this thing called a willingness to pay rate, where you sort of boil it down to being like, if the therapy is costing on the order of $50,000 to $100,000 for quality adjusted life year, which again is contingent on all these things, is generally recognized as a cost-effective therapy. But I'd say it's a complicated issue to be aware of. And so I want to finish up with a couple of slides to think a little bit about our institution and our institutional efforts around this. And I think when we talk about these therapies, we kind of can bucket them into three big categories as we think about sort of how we have to be thinking about them operationally. So there are therapies that are standard of care, and they carry their own sets of issues and regulatory stuff. But for you guys, I think acting more in the clinical trial space, um, there are many industry-sponsored studies available, and these studies, they bear similarity to other industry-sponsored studies that we participate in. But the nature of these therapies, I think, is so complex, and the toxicity profile is complex and significant that I think there are issues in this space that are a bit different than just doing drug-based studies. I will reiterate, though, that we've been leaders in the field. Manali was the lead author on Transform, and our team did a great work on it. And we were leaders in both the T-cell receptor therapy for sarcoma and the melanoma TIL, Teresa Medina and Bree Wilkie being the lead physicians on those projects. We put the first patient in the United States on a lupus nephritis CAR T trial. Amber Podol was is the PI of that. Larry Moreland in rheumatology put the first rheumatoid arthritis patient on a regulatory T-cell CAR T trial just about a month ago. So we're active in the space, and it's exciting. And then the other important thing to bear in mind is that we have this thing called the Gates Institute on campus, which is sort of the institutional academic arm of cell therapy led by Terry Fry. Had about a $200 million infusion a couple of years ago. And at the Gates Institute, we're able to manufacture our own CAR T cells and other cellular-based therapies. And we have internal product development going on and translational research. And the goal there is to make phase 1 studies with institutional science and be leaders in the field. The first thing we did at the Gates Institute was to just develop our own CD19 CAR, just to go through the complex rigmarole of the regulatory exercise of getting a CAR T into patients. There's nothing too special about it as compared to the other CD19 CARs. But the nice thing about having it is that we can then write clinical trials to use it in places where maybe others wouldn't or get ahead of the game. And so my colleague Mark Schwartz, one of our lead ALL doctor, has a CAR T trial using our institutional CD19, where we're giving CAR Ts to patients with ALL if they have any detectable disease after their initial therapy. So earlier in therapy, and if the results are as good as we hope they'll be, it could be paradigm changing for treatment of that disease. We also have a CD19 by 22, a bicistronic CAR that's in investigation, and then a pipeline of other products with an increasing pipeline of products also in the solid organ space. So, one of the great challenges here is this is highly multidisciplinary stuff, and we're at a highly complicated institution in terms of the different participating bodies. And so we're trying to get organized around that. Now, importantly and excitingly, the UC Health has recognized the importance of cellular therapy and its growth, and it's created 2 director-level positions, one occupied by Aaron Weston, who previously worked at cell therapies at Mayo in Arizona and at St. Louis, and worked in solid organ transplant here for a few years, but is now our director of operations and is great. And then Aaron Stoner, who's a long time nurse in the Transplant BD Blood Disorder Center and has had most every job, nursing director. And so they're kind of great resources for garnering resources from UC Health as we build this out. And I think we're thinking about the creation of an overarching cellular therapy medical director to sort of wrangle all of these different groups, with then in the different groups that participate, medical directors who will sort of be representative of their group. Now, again, this stuff is all changing, and it's all it's hard to plan for because of the rapidity with which it changes. But I think there's a general sense that many of these products share some commonalities that will benefit from some degree of centralization. And so once they become standard of care, for sure, all the regulatory requirements and so forth, I think, lend themselves to the notion of standardization. But even in the clinical trial space, I think there's things to consider there. And so I think that a general notion of a model to which we are trying to head is that there'll be a central resource that can help with a number of the things that are unique to cellular therapy, and that different groups who are involved in these cellular therapies will work with that central group. This is meant to be 100% collaborative, not patients are shipped to cell therapy and then cell therapy takes them and then sends them back. But that we all work together in this collaborative fashion with this central resource. And so we're in the process of trying to organize that and think about what it looks like. And again, particularly germane to you guys, I think in the context of clinical trials, we are struggling with and thinking about how to best support clinical trial research in this space. And I do think that some degree of a central resource to help where everybody can use it, but also collaborate is important. This stuff, again, it's coming fast and furious. There's a lot of people who don't know what they're doing on all sides of the issue, including the sponsors. And so we're sort of trying to work through this and come up with the best plan for it. The School of Medicine is standing up a data safety monitoring resource review committee for cell therapies. In the Cancer Center, we're way ahead of everybody else on campus around these issues. But folks in like rheumatology and nephrology, they don't have anything like PRMS, and there is no major data monitoring stuff. So we're looking to build that out and again, have it work for hopefully everybody effectively. So just to wrap up, I would say, exciting times. This is a broad, expanding, multi and cross-disciplinary field. We are anxious to all work together at the institution to develop a system that works effectively and well. But I think, again, based on my personal experience, sort of getting tied into these projects with people who have less experience or less knowledge, it works far more effectively if we have a central resource where people can go and we start there rather than just shotgunning everybody and pure chaos. So there's lots and lots of folks to thank for all that's going on. Highlighted a few of the folks here. And of course, thank to all you guys for all the work that you're doing in this space. So with that, I will wrap it up. I'm happy to answer any questions if there are any. And thank you all for coming and listening.
Thank you, Dr. Gutman, for a really great and informative presentation. We do have time for a few questions, so feel free to either unmute yourself or type your question in the chat. We are going to start with a question that was submitted ahead of our presentation today, so please bear with me, and I do apologize if I misinterpret this individual's question, but it's stated as, though these treatments hold a ton of promise, there are considerable cost and logistical challenges that you discussed briefly towards the end there in delivering these treatments to patients, on top of the astronomical fees that are charged when they are approved. So the question is, what do you see as a breaking point for a healthcare system in relation to cellular therapies, given the financial assistance burden, while others are still struggling to rebound from personal losses due to the pandemic?
Yeah, no, I think it's an important question, and I think we tried to touch on it a bit in the discussion. I think one of the things that I think is potentially also an even more significant challenge than these financial questions is that these are very complex, difficult therapies. And they, at least in their current forms, they often require patients to locate here for a month. And so in addition to basic financial issues, there's sort of socioeconomic and psychological barriers that need to be overcome that can be quite problematic. And it's not just as simple as like trying to do the right thing and saying that everybody should have access to these equally, like regardless of ability to pay, because the problem is that these can be potentially toxic, potentially lethal therapies. And if you're not in a position to be able to be supported through them appropriately, that can create a lot of problems too. So for both these issues, financial and sort of psychosocial economic support in general, I think we are hopefully going to be able to move towards the therapies being simpler, being shelf-based, being, and there's a lot of work going on in that space. However, I would also just, remind everybody that though the cost of one of these things is quite significant, I mean, $500,000 is no joke, it's a one and done therapy if it works. And so, the cost of cancer drugs in general these days is not going down. And the cost of care of a patient who is, getting chronic treatment for multiple myeloma or for these solid organ transplants. I mean, if you can do a one-time done $500,000 therapy, it's probably pretty darn cost effective. But again, it needs to be effective for that to work out. If everybody still relapses, you've only just sort of bought some time. But very important issues and ones that we're certainly struggling with.
Thank you, Dr. Gutman. We have a few more questions in the chat. The first being, at this time, are you able to treat a blood cancer patient with CAR T who also has a rheumatological disease?
So when studies are in the clinical trial phase, you obviously have exclusion and inclusion criteria. And generally speaking, the inclusion and exclusion criteria in trials would prohibit the concurrent use of the therapy. But now that there's FDA approval, you can start to be a little less strict about those things. I think I can't think of any patient that we've treated with a concurrent rheumatologic disease, but absolutely it could be sort of a 2-for-1 with the right combination of disease. And so, I think we'll continue to get that data. But for right now, the rheumatologic diseases are really being treated in clinical trial only, and the blood cancers are are now treated as standard of care. So you might get some of that overlap, but we haven't really done it per se to treat both things.
Great. Another question is, how do you manage the potential conflict of interest with all of the players? Citing a specific case of Jesse Gelsinger, who died from a gene therapy treatment.
Well, so again, I mean, these are not riskless therapies, and I think it's true, what is true about clinical trials in this space and research and the consenting of patients is really, it's the same as is true in any clinical trial and in any investigational therapy. And, patients are informed, and we do the best that we can around informed consent, but there are risks. It's just the scale of magnitude here is potentially different than with some therapies. But difficult issues, but I think everyone is trying to do the best they can with them, and that's sort of what we do.